Hi, my name is Tammy, and I'm a fourth-year undergraduate student studying public health and economics at the University of Massachusetts Amherst.
My name is Rachel, and I'm a fourth-year undergraduate student studying environmental science with a minor in natural resource conservation at the University of Massachusetts Amherst. Today, we will be speaking about the work that we've been doing in the lab of Dr. Laura Vandenberg, where we have been researching the effects of perinatal BVP exposure on the composition of the stroma of the mammary gland on mice in adulthood.
In our environment, we are exposed to numerous chemicals that can affect our health of our cells, tissues, and organs. Some of these chemicals alter the actions of our hormones in our body, and so are called endocrine disrupting chemicals, or EDCs. Butyl benzyl phthalate, or BBP, is one of these chemicals. BBP is a man-made common plasticizer found in cosmetics, adhesives, sealants, floorings, paints, coatings, and numerous plastic and rubber products. The most common use of BBP is to make manufactured plastics flexible and elastic, which seems like an advantage, but because it's not covalently bound to these plastics, BBP and other phthalates can be released from these products into our environment. Because of this, BBP and other phthalates are commonly found in soil, sediment, animals, food packaging materials, indoor dust, oils, and infant formula. It is also widely detected in human samples like urine. Prior studies suggest that BBP can cause reproductive and developmental toxicity. Phthalate exposures can lead to reproductive effects impacting the estrous cycle, including prolonged estrous cycles and missing ovulation in adult rats. This can decrease fertility, disturb pregnancies, and increase the risk of fetal death and teratogenic abnormalities in female rats.
Based on prior studies of BBP, the European Commission has already banned the chemical from children's toys, whereas regulatory agencies in the US have not taken actions to date. There is strong evidence indicating that BBP has endocrine disrupting properties, in part because it can disrupt function of hormone-sensitive organs like the mammary gland. BBP is known to be an estrogen receptor agonist, which means it mimics the actions of natural estrogens. It is also an androgen receptor antagonist, which means it can block the actions of androgens. The mammary gland is a hormone-sensitive organ that begins development in the prenatal stage and grows throughout lifetime during puberty, pregnancy, and menopause. It is a vulnerable organ for EDC exposures because its development is influenced and highly regulated by hormones such as estrogen, progesterone, testosterone, and growth hormone. Changes in the mammary gland tissue, including the expression of hormone receptors, immune cell populations, stromal composition, and metabolism features, can change the risk of mammary cancer. This means it is a good structure to study to determine how BBP acts as an endocrine disruptor and the extent of its effects. In vulnerable periods of development, disruptions to mammary gland development can produce long-term or even permanent effects, including delayed differentiation of ducts, retention of terminal end buds, and increased epithelial cell proliferation.
Our objective is to quantify the effects of perinatal BBP exposure on the composition of the stroma of the mammary gland in mice in adulthood. Using histopathological approaches and standard histology stains, we have quantified the number of mast cells and thickness of the periductal collagen and epithelial cells in the stromal compartment of the adult mammary gland. Additionally, immunohistochemical analyses were used to quantify the percentage of cells in the mammary epithelium expressing estrogen receptor, progesterone receptor, and Ki67, a marker of proliferation.
Here, we evaluated mammary glands from adult female mice that have been exposed to BBP via their mothers during the fetal and neonatal periods. Pregnant and lactating mice were exposed to BBP through an oil BBP mixture that was delivered orally via pipette feeding starting on day 1 of pregnancy and continuing daily through lactational day 21. The mice were administered an oil or one of 3 doses of BBP, 3, 500, or 18,000 micrograms of BBP per kilogram of mouse per day. 500 micrograms is the tolerable daily intake level established by regulatory agencies, and 18,000 micrograms is the no observed adverse effect level for mice. On lactational day 21, offspring were weaned. In this way, the F1 generation of mice was exposed to BBP through the exposed mothers, consistent with the entire in utero, gestational, and perinatal period. The F1 mice were weaned and housed until early adulthood. At 9 weeks of age, the left mammary gland was dissected from the mouse for morphological, histopathological, and immunohistochemical evaluation. So an ANOVA statistical test was performed against the control group. Let's look at a few results in more detail. So ER alpha is a marker of estrogen receptors, PR is a marker of progesterone receptors, and Ki67 is a marker of cellular proliferation. Through the use of immunohistochemistry, we quantified the percentage of mammary epithelial cells expressing each marker. In our result 1, immunohistochemical analysis did not indicate statistically significant changes in the percentage of cells expressing Ki67 or ER alpha between treatment groups. However, IHC analysis did indicate statistically significant differences between treatment groups for the percentage of cells expressing progesterone receptor. Specifically, these differences were revealed between the TDI and NOEL groups, as well as the TDI and 95th percentile groups.
The mammary gland stroma establishes the morphology of the epithelium. The stroma width influences the epithelial cell function of the duct. The epithelium and the stroma are important in embryonic development, ductal growth, ductal branching, and carcinogenesis. In our second result, perinatal exposure to BBP did not significantly affect the width of the periductal stroma and epithelial cell linings of the mammary gland ducts. Mast cells are one type of immune cell that is vital in allergic diseases, trauma, and neuroinflammation in the innate immune system. When inflammation occurs, the mammary gland stroma is affected, which changes the epithelium. In the mammary glands, mast cells promote ductal development, but they also promote angiogenesis and tumor cell proliferation. In our third result, perinatal exposure to BBP did not significantly alter the quantity of mast cells. Degranulated cells are when the cells are releasing pro-inflammatory mediators compared to granulated mast cells. The ratio of granulated to degranulated mast cells in the mammary gland stroma did not significantly change due to BBP exposure. Other outcomes we are evaluating include changes in the mammary gland tissue, including alterations to epithelium morphology, shape, and size of adipocyte, adipose cells, and others. Many of these features are risk factors for mammary cancer. Furthermore, disruptions to these outcomes can cause developmental issues, increase susceptibility to other environmental agents, or impair lactation and milk production. More data is needed to define the effects that BBP has on human development. Further research is also necessary to understand whether the effects of BBP on body weight that were observed in this study impact other metabolic outcomes or are relevant to BBP's mechanisms of action. Further work on BBP will examine the same outcome in the mammary gland at different ages and will also quantify the effects of BBP on the male mouse mammary gland.
We want to thank Klara Matouskova, who produced these animals and collected the mammary glands that we used in our study. We also want to thank Vandenberg lab collaborators, including Ruby Benzyl, Joe Jerry, Karen Dunphy, and Sally Schneider. Finally, this work was supported by a grant from NIH and the Commonwealth Honors College grant, and we are grateful for that funding. Thank you so much for watching.